Dzień dobry Państwu. Zaczynamy sesję nauk podstawowych dla chirurgów naczyniowych. Proszę Państwa, z nauk podstawowych właściwie bezpośrednio dla chirurga, chirurg nie korzysta z tego w sposób bezpośredni, no ale jednak wiedza na ten temat do czegoś prowadzi, rozszerza horyzonty, no, coś może jednak z tego również i w czasie wynika. Pierwsza prezentacja, first presentation, Lars Magdefessel, sorry, 20 minutes please.
Yeah, first of all, many thanks for the kind invitation and the kind introduction, I guess. I'm sorry, my Polish is not too good. I have a Polish postdoc, but he didn't really help me a lot last week. So, yeah, thanks for the kind invitation, and I'm happy to present to you today this presentation on microRNA-based therapeutics for vascular disease, which I guess for most of you will be a bit exotic. I will not be able to really show you any data on treatment options in human patients at this point. It's still at the experimental phase. But still, it's we, as well as some of the companies that are involved, are fairly excited about this, the potential therapeutic of using modulation of microRNAs to treat vascular diseases. Sorry. Yes, so this is something I guess everybody's very well aware of and everybody has been educated on. It's called the central dogma of life that has been proposed back in 1953-54 by James Watson and Francis Crick, that states that really all of our DNA gets transcribed into RNA and then further translated into protein. Well, we do live in very exciting times, so this central dogma of life has been challenged quite a lot over the last 10 to 15 years based on the decoding of the human genome and consortia work from Phantome and ENCODE and other forms of transcriptomic analysis. We really now do know that it's really only 2% of all of our human genome, of all our DNA that actually gets further processed into proteins. And the huge majority of our DNA gets processed or transcribed into something else, namely non-coding RNAs. And these non-coding RNAs include several subspecies. These so-called rRNA or tRNA are considered to be more housekeeping or functional RNA species, and long non-coding RNAs as well as microRNAs are considered to have an influence on gene expression, regulating gene expression, either on the transcriptional level, long non-coding RNAs on the transcriptional level, so-called epigenetic regulation, and this is still pretty much a black box. Nobody really knows what a lot of them are doing. And microRNAs and some subspecies of long non-coding RNAs have been shown to be these important translational inhibitors, so on the protein or on the messenger RNA level, by cleaving to messenger RNA, inhibiting the translation into proteins. And they have been very extensively studied over the last 15 years. So this is just a small definition about microRNAs. They're considered to be small non-coding RNA molecules, only up to 22 nucleotides large. They perform this post-transcription inhibition, and this is the way they are being synthesized, so that they are being transcribed within the cell nucleus out of regular DNA. Then they form this so-called double hairpin loop structure, and this very small form allows them to actually be exported out of the nucleus into the cytoplasm, which is very important because by being exported into the cytoplasm, they basically become a druggable target. They get further processed into mature single-stranded RNA, and then in this so-called RNA-induced silencing complex, or RISC, they bind to messenger RNA by complementary base pairing and silence their expression. It's believed that there's around 1,000 of these microRNAs existing in the human genome, regulating around 40 to 50% of the whole human genome. And they have been extensively studied for their potential of being used as therapeutic agents, not only in cardiovascular disease, but also in other diseases such as cancer. And the way you can therapeutically modify them is you can either use these so-called microRNA mimics, which you can show here in green, and their utilization is still a bit challenging because they're double-stranded, so you have to import them into the nucleus. But especially these so-called antimeres or antagomeres that by, again, complementary base pairing bind to mature forms of microRNAs within the cytoplasm have been extensively studied and are actually being used now in phase 1 to 3 clinical trials. apart from having an important function within the cell, microRNAs have also been shown that they can be exported out of the cell into circulation, which makes them basically available as biomarkers. And this has really become its own field of study, microRNAs as biomarkers, not only again in cardiovascular disease, but also in many others. There's different forms of how these microRNAs can be released into circulation. There's more active forms where they're being bound to exosomes or microvesicles. And some people or some researchers believe that they are actually very important for intercellular communications. Some have been shown that they are being released bound to just HDL particles, and they have been shown to play an important role in cholesterol homeostasis. And the huge majority, so 80 to 90% of all microRNAs are being released just bound to proteins, which is also important because the main protein they are being released with is ARGO2. And ARGO2 is a very important microRNA, mRNA binding protein. And this is usually the slide for all the clinicians, because as soon as they hear the word microRNAs, they immediately fall asleep. Because for them, microRNAs is still something that is very far on the horizon, that is very basic and not really has entered the clinic, which indeed is actually not true. So if you look at the NIH homepage, clinicaltrials.gov, you will see that there's 287 clinical trials currently involving microRNA modulation or looking at microRNAs for biomarker potential. And this image kind of lists the ones from the cardiovascular field that basically have been furthest advanced. So all the biomarkers are in blue that are currently being tested. All the treatment options are in green. And again, most of these have either already entered phase 1 clinical trials or are in preparation of entering small clinical studies. And really kind of like the furthest advanced ones, if you want to, like the poster boy of microRNA therapeutics, is the inhibition of microRNA 122, which is now FDA approved. So it's actually already in the clinic. You can use it to treat patients with chronic hepatitis C virus infections. It's been shown in phase 3 clinical trials that if you inhibit this microRNA, up to 90% of all patients are virus-free after 4 weeks of treatment. This microRNA 122 is necessary for the hepatitis C virus to replicate. And again, if you block it, you can get patients hepatitis C virus free. And then more from the cardiovascular field, at least one of them, inhibition of microRNA 21 has just been approved to treat patients with Alport syndrome. So Alport syndrome is a chronic kidney disease, fibrotic kidney disease. It's now, again, already approved to treat patients with this sub-disease of chronic kidney diseases. And targeting miR-103, 107 has been shown that it can be useful. And again, also now I think in the phase 3 clinical trial, AstraZeneca has stepped in, so Big Pharma is all of a sudden stepping in patients with NASH, so non-alcoholic steatohepatosis, in patients with prediabetes or type 2 diabetes. The diseases we've been most extensively studying over the past 5 to 10 years are abdominal aortic aneurysms, and this is a disease you already all are, I guess, very well aware of, maybe know much more about than I do. So just to give you a brief overview, it's considered to be a dilation of the infrarenal part of the aorta to more than 3 centimeters. It's a fairly common disease and lethal in case of weak root ruptures. The incidence, a bit depending on the study population and cohort you're looking at, is in 2 to 11% in men, but only 0.5 to 2% in women older than 65 years of age. There's some association with atherosclerosis-related diseases, but there's also some distinct differences. The risk factors, they certainly sound very familiar. So if advanced age, male sex, arterial hypertension, history of smoking is very interesting because more than 90% of a AAA are considered to have a history of smoking. And except for lung cancers, there's really no other disease that has such a high prevalence of smoking than AAAs. There seems to be some family history and genetic associations, and I think we'll hear some more about that later today. And diabetes, in this case, is actually a disease you would like to have, because diabetic patients, at least to a certain extent, have been shown to be protective from aneurysm disease. And of course, very important, there's no effective therapeutic strategies at this point for the early stages of the disease. So really the only treatment options we have is surgery or placement of an EVA prosthesis. The way we studied abdominal aortic aneurysms, again, we're fairly experimental heavy lab, so we used these 2 mouse models of aneurysm disease, the so-called allostase infusion model, where you do a local and chemic induction of an aneurysm, and if you wait for 28 days, you get a very histomorphologically very nice aneurysm in the infrarenal aorta. Or we use the angiotensin II infusion model, where you get a suprarenal aneurysm. It's considered to be more of a rupture or dissection model, not so much of an aneurysm development model. And of course, we also studied human AAA tissue from patients with different type of risk factors, different sizes of aneurysms, and compared that to control tissue from organ donor controls. And then to study microRNAs, we used microarray technology, which allows you to look really for all microRNAs being significantly dysregulated, in this case actually in the murine models of AAA disease. And the furthest upregulated microRNA on this array was microRNA 21. The one that was furthest downregulated is microRNA 29b. And another one based on its target profile, again, microRNAs have gene targets that they target, we believed was very interesting, was microRNA 24. And we became even more interested in these three microRNAs when looking at the gene profile. So we ran basically on the exact same samples, not only a microRNA array, but also an mRNA or gene expression array. And we did see that 80% of all targets or genes being dysregulated in mouse aneurysm tissue were predicted to be targets of these three microRNAs, again implicating that these three microRNAs must be very important for aneurysm development and progression. So what are these microRNAs doing, or how can you therapeutically modify them? I apologize in advance for this very busy slide showing the mechanisms of AAA development or the pathology behind AAA development. You can really pin it down to three main mechanisms based also on the cells or cell subtypes that are being involved, which are macrophages and lymphocytes. So you have inflammation going on in the vascular wall. You have dying or apoptotic smooth muscle cells, and you have an impaired matrix remodeling. So impaired collagen gene production and impaired fibrosis, which again also stabilizes or is supposed to stabilize the aortic wall. So what are these three microRNAs now doing? So if you overexpress microRNA 21, you can significantly induce smooth muscle cell survival or proliferation of smooth muscle cells. So all the yellow cells that you see in the below image, MIR21 induction, are smooth muscle cells. It acts through targeting a tumor suppressor gene called P10, which upregulates phosphorylation of AKT and inducing, again, survival of smooth muscle cells, stabilizing aortic walls. If you inhibit microRNA 29b, it's actually a downstream target of TGF-beta signaling, which has been shown to be very important in aneurysm development. And so if you downregulate microRNA 29b, you significantly induce collagen gene expression and other matrix components such as elastin and fibrillin-1, and you get a pro-fibrotic response in the aorta. And again, so all the yellow cells in this case, in this polarized light microscopy image, are collagen fibers. So if you stabilize the aorta with collagen fibers, you as well, of course, get smaller aneurysms compared to the ones that get a mismatch control in this case. And the third microRNA, so if you overexpress microRNA 24, you target a gene called chitinase 3-like-1, which is an important regulator of macrophage survival. So you will have significantly less macrophages in the aortic wall and significantly less inflammation. And by having that, again, you will induce smaller aneurysms with this microRNA therapeutic. One thing, of course, that is very important, one thing we did, which is what everybody in the field is doing, we performed systemic injections of these microRNA modulators. And of course, if you systemically inject something into a mouse, it's very nice that you can induce fibrosis in the aorta, but you will, of course, induce fibrosis in many other organs as well, especially in the ones where these microRNA modulators will assimilate to a much higher extent, such as the heart, the kidney, and the liver. And indeed, when we looked for collagen gene expression in these three organs, we did see that we upregulated collagen gene expression up to 40 and 50-fold, which would lead to cardiac fibrosis, liver fibrosis, and kidney fibrosis. So basically an impaired organ function, making this treatment for translational purposes in humans basically useless. And one idea we had to avoid these limitations or to still kind of push for these microRNA therapeutics was, and that's kind of like, yeah, the beauty of vascular disease, you can use local delivery mechanisms. You can use balloons and stents, and we've been experimenting with both, so using drug-eluting balloons as well as stents. So stents basically to anchor these stents and to avoid endoleaks, and drug-eluting balloons to treat the early stages of aneurysm development. And again, that's kind of like the stage we are right now. We look in large animal models, so land-raised pigs, so really huge pigs, where we again induce an aneurysm and then treat them with either a systemic injection of 29B or balloon-coated anti-29B, so a drug-eluting balloon. And we treat the aneurysms when they're at an early stage, so after 7 days in this pig model. And we can see that with the balloon-delivered 29B, we can basically completely halt aneurysm development in this model. Whereas with a systemic injection, it takes up to 42 days to see a minor effect compared to untreated control pigs. And of course, we look again for gene expression, on-target, off-target effects. We do see that in the aorta, we get a higher uptake or a higher effect on collagen gene expression. So collagen 3A1 is the most prominent collagen gene, not only in the human aorta, but also in the pig aorta. And with coated balloons, we get significantly more higher increase of collagen 3A1 in the aorta, whereas in the liver, we basically cannot detect any significant induction of collagen 3A1. And the second disease I just briefly try to cover because it kind of fits into drug-eluting devices to deliver substances to the vessel wall is instant restenosis. So again, a disease you all are very well aware of. It's defined as a 50% luminal narrowing after angioplasty. You kind of divide into early instant restenosis, which usually occurs after treatment with a bare metal stent after 6 months, or late instant restenosis after 6 months, up to 5 years with drug-eluting stents. The microRNA, in this case, we identified with profiling in human tissue samples, was microRNA 21, which you kind of already heard about, that it regulates smooth muscle cell survival and proliferation of smooth muscle cells. So we did check in a human biobank in patients with coronary artery disease and instant restenosis, compared that to patients who just had coronary artery disease. So these are tissue samples. And we did see that miR-21 is significantly induced in these patients who suffer from instant restenosis. So these were actually organ explants, again, patients who undergo heart transplant surgery. And then used in this case, a rat model of instant restenosis, where you denudate the endothelium in rats, and you put in a stent. And again, you can coat these stents if you want to, and which is something we did. So we used these non-polymeric translumina stents. We coated them with an anti-21, so an inhibitor against this microRNA. You can check with electron microscopy whether your stent is really well coated, and you can use fluorescent dye to actually label these integramers. You can study where they are being actually uptaken. And this is kind of what occurs. So if you look at the off-target effects, you do see that there's a slightly higher uptake in the drug-eluting stent group compared to the group that receives a bare metal stent and the systemic injection. And if you, again, follow basically the fluorescent dye with systemic injection and local delivery with the drug-eluting stent, you do see that you basically, the systemic injection ends up basically everywhere, preferably in the kidney and the liver, whereas with the local delivery, you don't see any uptake of these microRNA modulators after deploying these drug-eluting stents. And we, of course, do not only look at the off-target, but also the on-target effects. And we do see that with the local delivery, again, we basically have no instant restenosis after 28 days of treatment, whereas with a systemic injection, you get around a 30% instant restenosis, something you can also see if you perform OCT, kind of like the gold standard to measure instant restenosis. So again, with the anti-21 stent, you have a very few, so up to 10% myointimal hyperplasia, so proliferating smooth muscle cells, whereas with the bare metal stent, you get around a 30% instant restenosis, again showing that this treatment might be feasible for human patients with instant restenosis or recurrent instant restenosis. So in summary, I hope I was able to show you or convince you that microRNAs do play an important role, not only in AAA disease, but in cardiovascular diseases in general. For AAA disease, again, our 3 main microRNAs we're interested in is miR-21, which regulates proliferation and apoptosis of smooth muscle cells, miR-29B, which is an important regulator of extracellular matrix remodeling and collagen gene expression, and miR-24, which targets aortic vascular inflammation. On top of that, microRNAs are novel targets to prevent instant restenosis, and local miR-21 inhibition significantly limits myointimal hyperplasia. One important feature of miR-21, it's highly expressed in smooth muscle cells, but basically not at all in endothelial cells. So if you block something only in smooth muscle cells, but not in endothelial cells, the re-industrialization process in these miR-21 treated arteries will not be hampered, again making this a very feasible therapeutic target. Systemic microRNA modulation to treat vascular diseases is basically not feasible because you will have huge off-target effects in other organ systems where these modulators assimilate to a much higher extent, such as the liver, the kidney, and as well as the heart, making it, in our mind, very important to look for local or cell type specific delivery tools to use these microRNA modulators. And the last point, microRNAs can also be used as diagnostic and prognostic biomarkers for disease detection and prediction, something I did not talk about today, but again, microRNAs are also relatively stably released into circulation and have been shown to have an important value as biomarkers in patients with acute myocardial infarction, for example. We are looking at them as a tool to predict aneurysm growth, which is also fairly exciting. And with this last slide, I would just, again, first of all, like to thank the organizers for the kind invitation and everybody involved in our studies, people from our own lab, collaborators from the Karolinska Institute, vascular surgery, of course, very important. My previous lab at Stanford, collaborators there, Hamburg, Sonja Schrepfer's group, with whom we do the instant restenosis trials, our industry support, very important, of course, and all of our funding agencies. And thank you for your attention. I'll be happy to answer any questions afterwards, I guess.
Thank you very much. Jakieś pytania, głosy w dyskusji? Pani profesor?
Thank you. Nice talk. I have a question. I was wondering whether there are any data on the different type of microRNA involved in venous thrombosis or thoracic artery aneurysm not associated with Marfan syndrome or Marfan-like diseases.
So for thrombosis, yes, there's a lot of biomarker data. A lot of these microRNAs are highly expressed on platelets as well as other circulating cells like erythrocytes. There's also a couple of microRNAs that have been shown to target different coagulation factors, so they're also being studied as potential anticoagulants right now. For thoracic aneurysm, so out of these microRNAs that I've shown, only miR29B has been shown to be involved in, at least in Marfan syndrome. So in a subtype of thoracic aneurysms. It's the expression of these microRNAs between thoracic and the abdominal aorta is completely different. So which is kind of interesting. But again, the composition of matrix cells, smooth muscle cells, they come from different origin if you look at the thoracic aorta and the abdominal aorta, which kind of explains that. But 29B has been shown to be in Marfan syndrome to be involved.
Thank you.
Do you think it's possible that there are any adverse effects of using microRNA as a therapeutics?
Many. So yes, it's it's very dependent on the cell subtypes microRNAs are being expressed, of course, and how you deliver them. So I think systemic delivery of them will work for diseases where you try to trigger or to target the liver or the kidney, for example, because that's where all these microRNA modulators will end up. You always need to consider these off-target effects because microRNAs have hundreds of mRNA targets. And again, as emphasized, so I think for cardiovascular diseases, local delivery or cell type specific delivery is the only way that at least a handful of them might end up eventually in the clinic. But we're fairly close, I think.
Czy jeszcze jakieś pytania? Dziękuję. Thank you very much.
Thank you.
Optymalizacja doboru przeszczepów naczyniowych w pomostowaniu aortalno-wieńcowym. 5 minut. Bardzo proszę o czas.
Szanowni Państwo, bardzo dziękuję za zaproszenie do przedstawienia tych wyników. Są to wyniki, które powstały w ramach już blisko 5-letniej współpracy pomiędzy ośrodkiem badawczo-rozwojowym znajdującym się w specjalistycznym szpitalu we Wrocławiu. Tam jest realizowany projekt WROWASK oraz naszym uniwersytetem. Jestem przedstawicielem Katedry i Zakładu Histologii i Embriologii. Celem naszych badań było po pierwsze określenie optymalnego sposobu doboru przeszczepów naczyniowych w pomostowaniu aortalno-wieńcowym, to znaczy, które naczynie jest najbardziej odpowiednie do wykonania pomostu u konkretnego pacjenta. Cel drugi to określenie wykładników niepomyślnej prognozy funkcjonowania takiego przeszczepu. I cel trzeci to stworzenie programu w formie takiej aplikacji sieciowej, która byłaby programem wspomagającym decyzyjność lekarską w przypadku właśnie doboru takich pomostów. Chodziło tutaj o to, żeby te efekty naszej pracy laboratoryjnej przetłumaczyć na język zrozumiały dla osób pracujących w klinice i ten program już dzisiaj funkcjonuje. Na końcu Państwu przedstawię, gdzie go można znaleźć. Pacjenci, którzy byli objęci naszym badaniem w latach 2010-14, to jest ponad 750 osób w tej chwili. Od 450 osób była pobierana żyła odpiszczelowa, od 100 tętnica promieniowa i od 200 tętnica piersiowa wewnętrzna lewa bądź prawa. Z każdego fragmentu tych naczyń, z których był wykonywany pomost, pobieraliśmy fragment około 1 cm. To dzieliliśmy na dwie części, klasycznie na histologię i druga część do analizy RNA. I techniki, które były wykorzystywane, to pierwsza badanie przesiewowe, wytypowanie genów kandydujących w oparciu o mikromacierze ekspresyjne Affymetrixa, czyli prześledzenie absolutnie ekspresji wszystkich genów znajdujących się w danej próbce. Następnie te geny kandydujące określaliśmy ich poziom już ilościowy w technice RT-PCR i na końcu określaliśmy to w preparacie na poziomie białka. Jeżeli chodzi o wyniki, to możecie Państwo zobaczyć, że w zakresie genów tych, które zidentyfikowaliśmy, ich podwyższoną albo obniżoną ekspresję, to mamy grupę 6-7 genów różnych dla tętnicy i różnej dla żyły, które wskazywały nam albo pomyślną prognozę, czyli to, że ta tętnica będzie jako bypass funkcjonowała w sposób niezakłócony, czyli generalnie nie dojdzie do okluzji i tak jak Państwo widzicie, czynniki genetyczne, które były związane z okluzją tejże żyły w okresie pierwszych 12 do 18 miesięcy. Ponieważ wiadomo, że ten wynik w aspekcie genów jest trudny do powtarzania w lokalnych na przykład laboratoriach, w związku z tym w drugim etapie przeszliśmy na poziom białka, żeby zobaczyć, ekspresja których z tych genów jest również zachodzi na poziomie białka i jakie to ma znaczenie prognostyczne. I tutaj wyznaczyliśmy w przypadku pomostów tętniczych ekspresja dwóch białek PDGFA i TIMP2 jako wskaźniki pomyślnej prognozy, natomiast recepty dla NGF, receptor i TNFR były to wskaźniki niepomyślnej prognozy w przypadku pomostów żylnych. I teraz wszystkie te wyniki, które mieliśmy, przetłumaczyliśmy to na współistniejące choroby, poziom glukozy, lipidogram, masę ciała, wiek i płeć danych osób i w efekcie stworzyliśmy program, który jest programem możliwym już do zastosowania w klinice. Ten program na niebiesko macie Państwo podkreślony, to jest aplikacja znajdująca się pod adresem ebypass.pl, pisane z myślnikiem albo bez myślnika. Jeżeli Państwo na tę stronę wejdziecie, do czego bardzo zachęcam, mamy w tej chwili około 100 zarejestrowanych użytkowników. W ramach tego programu możecie Państwo zobaczyć, w jaki sposób system podpowiada lekarzowi, jakie naczynie wykorzystać u konkretnego pacjenta do bypassu. Dziękuję bardzo.
Serdecznie dziękujemy i gratulujemy. Gratulujemy również czasu. Bardzo proszę, jest czas na pytania. Pani profesor?
Rozumiem, że także przy wczesnym zamknięciu, prawda, w tym systemie nie uwzględniamy na przykład ile chory ma płytę krwi, jakby jakości krwi innej niż glikemia i lipidy, które zwykle są jednak kontrolowane, no przynajmniej względnie dobrze nie uwzględniamy. Nie wydaje się Panu, że choćby nieuwzględnienie płytek może być, nawet liczby płytek, pewną wadą?
Braliśmy to pod uwagę, jednakże na etapie konstruowania skonstruowania samej aplikacji nie wychodziły nam tutaj zależności, które umożliwiłyby wciągnięcie tego do całego algorytmu. I tylko braliśmy te elementy, które jednoznacznie nam na zasadzie korelacji wskazywały, że to może być przydatne. Natomiast...
Na ilu osobach to nie wychodziło? Z ciekawości zapytam.
750 osób.
Dziękuję.
Mieliśmy 750 osób. No, trochę więcej, ale generalnie to byli ci pacjenci, którzy na końcu, do samego końca byli analizowani. Natomiast mamy całą grupę chorób współistniejących, które również odhaczamy, odpowiednie okienka i wtedy na bazie tych współistniejących chorób to też ten algorytm odpowiednio nam podpowiada. Także zachęcam do spojrzenia w program i zgłaszania jakichkolwiek uwag. Również to jest pierwsza wersja. Mam nadzieję, że będzie ona mogła być wdrożona na szerszą skalę.
Dziękujemy za bardzo, bardzo ciekawy referat.